IB Bio, Option B, Biotechnology, Bioinformatics, Part 3, is the first of two movies that look at B.2, Biotechnology in Agriculture. The primary focus of Part 3, B.2A, this movie, will be aspects of transgenic techniques that set the stage for a close look at applications of transgenic techniques in agriculture. That'll be Part 4. The essential idea is crops can be modified to increase yields and to obtain novel products. Here is the outline of all the movies available for Option B. Use this outline to find the movie you need for review. This movie is focused here. Transgenic organisms were first reported in the 1970s. Glowfish, seen on the left, were the first genetically modified organisms sold as a pet. Glowfish have a jellyfish gene that produces a protein that fluoresces. The Flavor Saver tomato on the right was the first genetically modified organism sold commercially in the 1990s. The Flavor Saver tomato produces a protein that interferes with natural decomposition. Thus, the Flavor Saver tomato has a longer shelf life after having been picked from the vine. Transgenic organisms produce proteins that were not previously part of their species' proteome. Golden rice, seen on the left, was genetically modified to produce vitamin A. BT corn on the right has a gene that produces a protein that is distasteful to the corn borer pest. The production of this novel protein in corn has increased yields. Genetically modified crop plants can be used to produce novel products. Here are two IB syllabus statements that come from the biotechnology unit that you've previously studied. State that genetic modification is carried out by gene transfer between species. Outline a basic technique used for gene transfer involving plasmids, a host cell, restriction enzymes, and DNA ligase. From your earlier study, you should remember that the genetic constitution of bacterial cells includes one large circular chromosome and numerous plasmids. Plasmids are small circular rings of DNA that have become an important tool in biotechnology. Plasmids are used as vectors in the creation of transgenic organisms. This is a micrograph of a plasmid. The genes of one organism can be successfully inserted into the genome of another because the genetic code is universal with minor exceptions. When genes are transferred between species, the amino acid sequence of the polypeptide is unchanged. Plasmids, seen here, can replicate independently of the primary bacterial chromosome. Plasmids are circular rings of DNA without histone proteins. As well, plasmid base sequences can be cut by enzymes known as restriction enzymes. The cut sites allow a foreign gene to be inserted. The restriction enzyme cuts the DNA at a precise location in the genome. The enzyme cuts at a precise location, a precise sequence of bases. This opens the plasmid to make it ready to accept another piece of DNA from a different organism. Here is a diagram of a plasmid cut with a specific restriction enzyme known as EcoR1 that creates complementary ends on the cut site. These sticky ends, as they're known, allow another gene with the same complementary sticky ends to be inserted into the plasmid. This diagram is similar to the last one. A restriction enzyme known as EcoR1 has cut the plasmid at a specific site known as a restriction site. The restriction enzyme opens the plasmid, leaving two ends to which new DNA from a different organism, could be inserted. The restriction enzyme opens the DNA in two locations, and this section of the DNA can be removed. Then a new section of DNA can be inserted. This gene of interest right here has the same sticky ends because the DNA of both species was cut with the same enzyme, and DNA ligase forms covalent bonds between adjacent nucleotides here and here. So we can start with a plasmid and DNA from an organism of interest, let's say a virus of interest, and this would be the target gene. Imagine that this viral gene produces a protein which can be collected and injected into people as a vaccine. In other words, the protein resulting from this gene stimulates the body's immune system to produce memory cells, and as a vaccine, we need a lot of this protein. Thus, what we need is a host organism for the gene, and we need a vector to carry the gene into the host. And with this, we could have industrial production of the protein. Now, both the plasmid and the DNA of interest are cut with the same restriction enzyme. Each DNA molecule has the same sticky ends. The target gene joins the plasmid, the sticky ends have complementary base pairing, and ligase seals the gaps between nucleotides here, 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 and here. And then the recombinant DNA molecule, the recombinant plasmid, is inserted into a cell, in this case a bacterial cell. The bacterium is known as Agrobacterium tumefaciens, and this bacterium can be used to infect plants, which would industrially produce the protein from the target gene seen here. Here, along the left, is the sequence of steps that I've given you for forming a transgenic species. In this diagram, we have a viral gene inserted onto a plasmid that is introduced to a plant using a bacterium that infects plants as a secondary vector. The plant that receives the gene is, in this case, soybean. The transgenic plant makes protein, not a soybean protein, but a protein native to the virus from which the inserted gene has come. The protein can be used as a vaccine. The protein is purified and distributed to people in need. The soybean is a host organism grown on an industrial scale. The plasmid is the vector to carry the gene into the host. With this, we can have industrial production of the vaccine. As a note, the soybean in this example is a transgenic organism that produces a protein not previously part of its species proteome. A recombinant plasmid can be introduced into plants as the indirect vector. In this example, the bacterium, Agrobacterium tumefaciens, takes up the recombinant plasmid. The plasmid has a gene that will allow the plant to resist a herbicide. The bacterium infects the plant, causing a tumor to grow. The recombinant plasmid is introduced into the plant's genome. Let me say that again. The recombinant plasmid is introduced into the plant genome. Here is a relevant IB syllabus statement. Recombinant DNA can be introduced by direct physical and chemical methods or indirectly by vectors. There are plasmid vectors, as I've described already, and virus vectors, which I will touch on momentarily. Plasmids and viruses introduce target genes to new host cells in an indirect manner. Now, there are direct methods of introducing recombinant DNA to new host cells. Chemical methods include calcium chloride treatment or liposomes. Physical methods include electroporation, microinjection, and biolistics, the gunshot method. Recombinant DNA can be introduced by indirect methods, plasmids or viruses. In this image, we can see a target gene introduced to a virus that infects a cell. The virus delivers the target gene to the cell. The cell would then express the novel protein. For example, tobacco mosaic virus could be used to deliver a gene that produces Hepatitis B vaccine. Again, in this diagram, you can see a virus that has had its genome genetically modified with the target gene is inserted, the virus infects a cell, delivering the target gene. In this image, the virus has had a new gene inserted into its genome. The virus infects a cell, delivering the target gene to the cell nucleus. The cell is now genetically modified, ready to produce novel protein products. The tobacco mosaic virus, seen here, has its genome modified to include a gene that produces a protein serving as a hepatitis B vaccine. The virus delivers the gene to the tobacco plant that the virus infects. The tobacco plant, seen here in the lower image, is infected with the tobacco mosaic virus. The virus delivers a gene that produces a protein that is used as a vaccine against the hepatitis B virus. The plant mass produces the hepatitis B vaccine. Now remember, recombinant DNA can be introduced by direct physical and chemical methods. Chemical methods include the use of calcium chloride or liposomes, and physical methods include electroporation, microinjection, and biolistics, the gunshot method. Let's take a look at all 5 methods. In this diagram, we can see the direct uptake of DNA fragments using calcium chloride to sufficiently weaken the membrane for successful transformation. The target gene is incorporated into the genome of the host cell. Calcium chloride with a heat shock allows the direct uptake of DNA fragments, as seen in the last image. Calcium chloride also serves to encourage the uptake of recombinant plasmids so that we have transformed bacteria that can be identified and used to industrially produce proteins. Liposomes are phospholipid membranes that enclose DNA and deliver it to the host cell. The liposome easily fuses with cell membranes to deliver the DNA to the host nucleus. Here we can see the liposome having merged with the host cell's membrane to deliver the DNA to the host cell interior. So we've been looking at how recombinant DNA can be introduced by direct chemical methods, calcium chloride treatment, or liposomes. Now let's direct our attention to the physical methods, including electroporation, microinjection, and biolistics. Electroporation is the exposure of the cell culture to a high voltage electric pulse. Here is a cell before the electric pulse. You can see the DNA outside the cell membrane. The electric pulse causes pores to open in the cell membrane, allowing the uptake of DNA so that the cell becomes a recombinant cell. Here's the cell after the pulse with new DNA within. In this image, we can see the pore that opens up with the electric pulse. The opening allows the uptake of recombinant DNA. Electroporation is fast, inexpensive, and works with various cell types. Large numbers of cells can be treated simultaneously. Microinjection is when recombinant DNA is injected directly into the nucleus using a microneedle, as can be seen here. Here we can see DNA being injected directly into the nucleus. Microinjection requires highly trained personnel and is used when there are limited numbers of recipient cells. Here we can see the microinjection of DNA into the nucleus of an embryo, resulting in a transgenic mouse. In this diagram, DNA with a gene for an insulin-like growth factor called IGF-1 is inserted into a pig embryo. The transgenic pigs produce less fatty muscle. And lastly, among the direct methods that physically introduce recombinant DNA into host cells, we have Biolistics, the gunshot method. This is known as a gene gun that directly propels DNA into plant cells. A gene gun delivers a particle of heavy metal coated with plasmid DNA. In this diagram, we have DNA-coated gold particles fired at plant tissue. Through the last 8 slides, I have addressed direct physical and chemical methods of introducing DNA to cells. The chemical methods include the use of calcium chloride and liposomes, while the physical methods include electroporation, microinjection, and biolistics, the gunshot method. So all of this raises the question, how do we know when host cells have taken up the target gene? And thus the next IB syllabus statement. Explain how marker genes are used to indicate successful uptake. And that brings us to define the term marker gene. It's a gene used in biotechnology to determine if a nucleic acid sequence has been successfully inserted into an organism's DNA. So again, how do we know if the host cell has taken up the target gene? In the process of building transgenic species, the target gene is inserted into host DNA alongside a gene known as a marker gene. One type of marker gene would be a gene that confers antibiotic resistance. So if the target gene has been inserted successfully into host cells, the host cells with the target gene would also be able to grow on an antibiotic medium because the plasmid containing both the target gene and the marker gene for antibiotic resistance would be present. Any host cell that has not successfully taken up the plasmid would not be able to grow on the antibiotic. All of these plants are transgenic. Transgenic plasmids are constructed to hold the target gene and a marker gene, in this case, a gene that provides antibiotic resistance. Other marker genes might be present as well. As a note, the target gene is linked to other sequences that control its expression. For example, there must be a promoter. The promoter is a sequence of bases that allows RNA polymerase to bind to the gene for successful transcription. Keep this in mind. A promoter sequence must also be included in the construction of the plasmid vector. The target gene is seen here, noted as an open reading frame. The open reading frame is a sequence of bases that code for a polypeptide. When target genes are inserted into plasmids, other sequences are typically linked to the target gene to control the expression of the target gene. The promoter is an example of an upstream sequence that controls the expression of a target gene. More on this later in the movie. The bacterial colonies seen growing on the agar in this image display successful uptake of a transgenic plasmid. There were 2 marker genes on the plasmid taken up by the bacteria growing on this plate. One marker gene provided resistance to ampicillin. The second marker gene originated with a jellyfish and provides the bacteria with the capacity to fluoresce. The gene for green fluorescent protein, GFP, is a base sequence just under 750 base pairs. The GFP protein is composed of 238 amino acid residues. GFP exhibits bright green fluorescence when exposed to ultraviolet light. In this way, it is known that successful plasmid transfer has occurred. The gene for GFP has been successfully transferred to mammals, among many other species. You may question the ethics of transgenic species, and that's fair enough. That said, the scientists who first developed the GFP gene as a marker in the building of transgenic species were awarded the Nobel Prize in 2008. So let's move the conversation forward by connecting the dots among concepts presented in this movie. The concepts to be connected are target genes, upstream regulatory sequences, and open reading frames. All of these terms have come up earlier in this movie. So here's a relevant syllabus statement. Define the phrase open reading frame. An open reading frame is a sequence of successive nucleotide triplets that are read as codons specifying amino acids. Open reading frames begin with an initiator codon and end with a stop codon. An open reading frame is a length of DNA that contains sufficient nucleotides to code for a polypeptide chain. Open reading frames are the target genes used in the building of recombinant DNA. Here is a chromosome, unwound. You can see nucleosomes here, and a stretch of simplified DNA with an open reading frame, a start codon and a stop codon. An open reading frame has the potential to code for a polypeptide. An open reading frame is a continuous stretch of codons that do not contain a stop codon. Scientists hunt for open reading frames to locate genes. In other words, scientists look for long stretches of DNA without stop codons as an indication of the presence of a gene. Open reading frames are important to scientists working in bioinformatics. In this image, a gene is transcribed, then the mRNA is translated into a polypeptide. An open reading frame has the potential to code for a polypeptide. An open reading frame is a continuous stretch of codons that does not contain a stop codon. Get to know these three codons as stop codons. An open reading frame is successive nucleotide triplet codons that begin with an initiator codon and end with a stop codon. You can see the start codon here with AUG on the mRNA. The stop codon is here. Depending on your source of information, open reading frames will be referenced in DNA or RNA language. Be flexible and smart as you review this material. Open reading frames constitute the target gene in the building of transgenic organisms. Scientists hunt for genes by looking for long stretches of DNA without stop codons. The exact codons of a gene may not be known, but a long stretch of DNA with no stop codons would be an open reading frame within which there may be a gene. Now in this image, you can see the promoter here. RNA polymerase binds to the promoter. The promoter serves to regulate the expression of the open reading frame. The target gene is linked to other sequences that control its expression, and the promoter is the best or the simplest example of an upstream sequence linked to the target gene that controls the expression of the target gene. In this slide, the open reading frame is stated in RNA language. The open reading frame begins with a start codon and ends with a stop codon. Here is the transcribed segment of DNA. This is the gene, oriented 3 to 5, because the mRNA is transcribed 5 to 3. There are sections of DNA upstream of the gene and downstream that ultimately are not translated. The open reading frame, seen in red here, begins with a start codon and ends with a stop codon. There are upstream sequences linked to the open reading frame that control the expression of the open reading frame. In this image, you can see the target gene indicated as an open reading frame. Restriction enzymes XBAL and PST1 cut the plasmid and the DNA of interest in the same locations, creating sticky ends that allow the target gene, the open reading frame, to be inserted into the plasmid. This plasmid has a gene for factor IX, a protein involved in the clotting of blood. Individuals with certain genetic forms of hemophilia require factor IX to ensure that blood is clotted upon injury. Now, the gene is inserted into the mammary cells of sheep or cattle and the upstream sequences linked to this gene include a promoter called beta-lactoglobulin that ensures that the gene is transcribed in the mammary glands along with milk production. In other words, factor IX is released with the milk of the animal and is separated from the milk and purified to be made available to certain hemophiliac patients. Also notice the Tata box. The Tata box is another upstream sequence that serves to regulate expression of the target gene. In this diagram, you can see the open reading frame, also known in this context as the target gene. Expression of the target gene is regulated by upstream sequences to which the target gene is linked. As genes are manipulated and inserted into plasmids, it's important that regulatory sequences be included. And so, with this IB syllabus statement, I've brought the movie full circle to the place I started on slide 3. State that transgenic organisms produce proteins that were not previously part of their species' proteome. And I've defined the term proteome for you. Proteome is the entire set of proteins expressed by a genome, cell, tissue, or organism at a certain time. A proteome is the set of expressed proteins in a given type of cell or organism at a given time under defined conditions. For example, a human gene for insulin is inserted onto a plasmid that's come from a bacterium. The recombinant plasmid is inserted into a bacterial cell that produces insulin as a protein. This transgenic bacterium now produces a protein not previously part of its species' proteome. As well, the bacteria can be industrially cultured in a fermentation tank to produce industrial-scale quantities of the protein. And that brings us to the end of IB Bio, Option B, Biotechnology, Bioinformatics, Part 3, also known as B.2A.